What is in vitro physiology or patch clamp, and why are these techniques important? The brain consists of billions of brain cells called neurons, and the most interesting thing about these is that they produce electrical events called action potentials. Information from the outside world, like something you see or hear, is coded in trains of action potentials inside the brain. And these trains of information, like trains of ones and zeros in a computer, are sent back and forth between various brain cells through cables called axons. And this information is processed and integrated with each other such that at the end of the processing process, you understand what I'm saying to you, for example. So naturally, neuroscientists are interested in studying how electrical information is created and how it is flowing in the brain, because it's the language of the nervous system. And understanding that language means we're a whole step closer at understanding how the brain works. So one way to study electrical activities in the brain is by using sections of brain tissue instead of whole brains. So neuroscientists can take a brain out of an animal, for example, and section it into slices that are about 1/4 millimeter thick. Importantly, these slices are still alive and produce all the same electrical events that they would in the natural brain. And once those sections are prepared, they are put in some sort of tissue chamber and kept alive for up to several hours, while the neuroscientist can then study the electrical events in these pieces of tissue. How would one study electrical events? You use electrodes. An electrode is typically a glass pipette that is drawn to a very fine tip, and then it is filled with salt water for electrical conductivity. And there is a wire in the back of the pipette, and the electrical events that are recorded by this pipette are then sent to an amplifier and a computer for analysis. So once the brain tissue is in that slice chamber, in that holding chamber, this whole chamber can be put under a microscope for imaging of the cells, and then scientists can see the brain cells on a big screen in real life. And then they try to electrically connect with one brain cell they choose with that glass pipette to record electrical activity in there. And that's a very handy and very important way to study electrical activity in the brain, because you can see the brain cells, you can see if they're healthy or not, you can see which cell you're recording from, and one can also manipulate the environment inside the cell and outside the cell relatively easy, which would be a lot harder to do when one worked with the whole brain. So slice physiology is a very, very important technique in neuroscience, and patch clamp is the technique of recording electrical activity from a single brain cell with one of those glass pipettes.